Thank you, Hua. Good morning, everyone. My name is Jing Zhou. I am an associate professor in the Department of Biostatistics at UCLA, and I was a doctoral student of Ken Lin and Jenny Sinsheimer before, and I'm also an alumnus from the Department of Biomathematics. Now it's called Computational Medicine. So it's my great pleasure to introduce our first speaker today, Dr. Nancy Zhang. And Dr. Zhang is a professor, is a Gerli and a Linzhao professor of statistics at Wharton School at UPenn. And she also serves as a vice dean of the Wharton Graduate Division. Her research pushed the boundary of statistics, genomics, and computational biology, and she developed many influential methods for analyzing large-scale genomic and biomedical data. She has published extensively in top-tier journals, including Nature Methods, Nature Biotechnology, PNAS. And she also received numerous awards, such as Sloan Fellowship and the Medallion Lectureship from the IMS. Dr. Zhang also serves as the 2023 president of ASA session on statistics in genomics and genetics. And she also pushed forward the first Staging conference last year. So please join me in welcoming Dr. Zhang for her presentation. Thank you.
Thank you very much. Thanks, Jen. And it's just a great honor for me to be here in this symposium celebrating Ken's career, especially given everything that's been happening at LA, that from the East Coast, we can only see from afar. I'm just really happy to be here to celebrate with you. Ken, I met Ken first when he was at Stanford for his sabbatical, I think. It was in the year 2007, 2008, around that time. And I just started then as an assistant professor at Stanford. And at that time, Ken's work in genetics is already an inspiration for me. But what really helped me going forward in my career is a course that Ken taught when he was on sabbatical at Stanford. It was a course on the algorithm and optimization. And that was my first time learning about that material. Ken made that material not just beautiful, but also very practically useful. I still have his lecture notes in a binder on the shelf next to my desk. And many times, I think, after that course, in my research, I found myself actually going back to those notes to reuse some of the intuition and ideas from that lecture. It had a profound impact on me. So thank you to the organizers for putting up this event and giving me this opportunity to give a talk here. And thank you, Ken, especially for everything that you've meant to me in my career. So I'm going to share with you some work over the last many years. I've started this work before COVID. It's on single cell data integration. I think single cell may not be new to most of you. You've heard of the term, but what is single cell integration? So let me just start off with a quick introduction. So during the last decade, it's not an exaggeration to say that biomedical research have been driven by these single cell atlases that try to profile the variety of cell types, the variety of the trillions of cells in a human body, which are arranged in these tight-knit communities that give our tissues their function. For example, if you were to look under a microscope, this is Codex data, that type of protein multiplex imaging data of the human colon, you will see different cell types light up with different colors. They are arranged in very regular patterns, and these are crypts in a colon. But every tissue in the human body is made up of these tight-knit communities. And we're just starting to learn about how these cells change, their roles in disease, and their roles in for example, various human aging or as such. I'll give you some examples, okay? So what is cell integration? This talk will go through two running examples. The first example is a study of the colon that was conducted at Stanford. Here you start with fresh frozen colon tissue from a human donor. And this is from Gary Nolan's lab at Stanford. What they did was they took these 3 separate slices of tissue and put them through different technologies. The first one is this codex imaging, which gives you the spatial coordinates of different proteins that are cell type markers. And based on these proteins, we can map out where the different cell types are in the tissue and their spatial relationship to each other. The other technology is single nuclei RNA sequencing, which profiles the entire transcriptome over the 50,000 cells that they were able to harvest. And then the third technology is single nuclei ATAC sequencing, that is profile the chromatin accessibility landscape of a different sample of cells. So this is 50K cells, but it's not the same 50K as these others. So that was the data, but that's not ideal. What is ideal would be if we could have everything measured on the same cells mapped in space. So we would like to have not just protein, but RNA and ATAC all on the same cells in the same space. But there's no technology right now that can give us everything to this resolution. So that's one example. Another example is a study that was conducted and that's still going on at UPenn. Is my collaborator Caitlin Sessa's lab. They are studying chronic kidney disease. Okay, so there's already a very comprehensive single-cell atlas of the human kidney, right? We know all the cell types in the kidney, but we don't know how these cell types, what's their role in chronic kidney disease. So what they did was they collected data from 17 healthy individuals and 19 diseased individuals. And then depending on whether the sample is fresh or frozen. If it's fresh, they can dissociate the cells and put them through single-cell RNA sequencing. If it's frozen, then they have to do single nuclei RNA sequencing because they can dissect the nuclei, but not the entire cells. Okay, so some samples they have one, some samples they have the other. And if you were to take the 200,000 cells that were in total harvested in this study and put them on a low-dimensional embedding, such as a UMAP, which is a common embedding that is used in the field, you will see that they sort of, they look like that. And then if you were to color the cells as to which sample they come from, control versus disease, that's what it looks like. And then their goal of this study is to identify the disease-specific cell states, and also to identify the pathways that are changed in a cell type specific manner in disease. Okay, however, as I noted, there's 2 different protocols being used, single cell and single nuclei, depending on whether the sample is fresh or frozen. If you were to color the cells instead by the protocol, you see that the protocol completely separates the cells. Okay, and that's not surprising at all because, there's so much differences in not only in the technique, but also just there are different populations of RNA. One is RNA that's in the nucleus, one is in the whole cell. If you were to take these cells and color them by sample, so let's take a look. Okay, that's by protocol. That's by sample. You see that even within protocol, the cells separate by sample. And that just depends on how long that sample rested on dry ice, who was doing the experiment, the details. And this is called batch effects, right? There's a lot of statistics studies of batch effects. So here, instead of integrating across modalities, we would like to integrate across batches to remove the technical differences, to reveal the biological differences. Okay, so I'm just giving you a preview, right? So if you were to take this data and do what we call single-cell data integration using CellANOVA, which is the method we developed, you will get these clusters again, but they're sort of bigger clusters. And what we hope is that these clusters to represent different cell types, okay? Okay, so just a summary, there's two different setups with different goals. That was the kidney atlas. And then I also showed you the human colon atlas. Even though we're calling them both integration, right, cell integration, they're very different mathematical problems, right? So in the kidney atlas, we are actually having samples with different biological contexts, disease severity, sometimes treatment, and they're from the same modality. They're all measuring RNA, okay? And then in this setting, there's oftentimes many samples, tens, hundreds, and thousands of samples that are now being collected in studies of disease. And our goal is really to identify biological differences between these samples, okay? In comparison, in the colon atlas, we actually have the same tissue, right? Healthy colon from an individual, but we're putting it through different modalities, right? Measuring different things. They're different things on the same tissue, but different cells, right? Because once you burst open the cell, that's it, right? So our goal really is to construct a unified representation of this tissue, hopefully aligning the cells between modalities. Okay, so those are two different integration scenarios. They're sort of on the opposite ends of the spectrum. Okay, so what makes an integration scenario hard? That depends on two things. Okay, first is the strength of the feature linkage. And then what I mean by that? Okay, so let's go back and look at the colon example, right? Here, let's say that let's forget about ATAC-seq for now and just look at RNA-seq and protein, right? So RNA-seq, you're measuring the whole transcriptome, but for the codex of proteins technology, you're only measuring 47 proteins. 47 proteins, those are the features that are linking together these two modalities. And we know that there's actually a lot of differences between RNA and protein. The correlation is oftentimes not that high, right? So then this is the case where you have weak linkage, okay? How about ATAC-seq and RNA? ATAC-seq is 200,000 peaks. It's full, the full genome, right? So you have more features to link with RNA. However, there is also a very weak correlation oftentimes between the chromatin openness of a gene and how much of it is expressed. Okay, so that's better, but it's still pretty difficult. Now let's look at the kidney atlas example. Here we have single cell RNA-seq and single nuclear RNA-seq. They're the same modality, but two populations of RNA within the same cell. So it's easier. It's stronger linkage, okay? But that's not as good as if you had single nuclear RNA-seq on both samples, right? So, so the strength of feature linkage is the #1 consideration about how hard an integration problem is. The other thing that we need to consider is the strength of the biological signal, which we don't actually know, right? So it depends on the goals of the study. Let's take, for example, so it goes from weak to strong. Let's take, for example, this colon atlas study, okay? So the goal here is to just do a integrated profiling of the colon. And let me just give you a preview. If we were to apply what we call MaxFuse integration, which is a method we developed to it, and each dot here is a cell. This is a UMAP embedding, okay? So each cell here is colored by the modality, whether it's protein, RNA, or ATAC. So you see, good, it's, the different modalities sort of merge together within the same clusters overall. They still separate a little bit, but they merge together. And if you were to use gene markers to annotate these clusters, you will know that these are goblet cells, these are smooth muscle cells, these are the immune cells. So it's nice that the different cell types are separating out in this common embedding across modalities, right? So for example, we think it's a success that the macrophages are separate from the T cells, okay? But that's actually not that impressive because macrophages are big cells. They're very different from T cells. They have completely different biological characteristics. So here, I'm just giving you an example that separating out macrophages from T cells is considered very strong biological signal. So, it wouldn't be hard if you were integrating single nuclear RNA-seq and single nuclear RNA-seq, okay? Another example is the kidney atlas preview. If we do integration, we get this kind of UMAP where the clusters can now be annotated, right? They are annotated based on cell type markers. And then good, we are having a big cluster here that are the proximal tubule cells, which are the main cell types in the kidney. So that's not surprising that we found it because they are well studied, well known. But my collaborator wants to figure out how these cells change between control and disease. So how, are there subtypes of these proximal tubule cells that jump out during disease? You don't see it very well, but that is actually a very hard problem. Okay, so separating out within the same cell type, the different subtle states that relate to disease is really difficult. Okay, so now hopefully I gave you the sort of a setup, a quick intro to this subject. So this talk is about two different attempts at this problem over the last few years. I'll first, give you a very brief sort of look at the strong linkage scenario. So this is the kidney atlas example where we have many samples of the same modality comparing biologically different samples. And there our goal is to remove what we call batch effect. Then I will look at the other extreme, which is when you have weak linkage. So spatial protein data with single-cell RNA-seq data. This case, there's the linkage is so weak that our goal is just to identify the major cell type clusters. Okay, so let's first just look at the strong linkage scenario. So back to this kidney atlas, okay, example. All right, so here again, I have the cells colored by sample ID, and I can just tell you that when we first got this data from our collaborator, they've already tried all the different integration methods out there, for which there's like dozens of integration methods. And here is an example of what you get with one of those integration methods called Harmony. If you do single cell sequencing, you know about this method. It's very popular. Okay, Harmony has about 5,000 citations despite only being a few years out. And its competitors like Seurat, which is also a big name in this field, that has like 12K citations. So you see that this is really the bread and butter of many single cell studies. You can't analyze cells from across samples without first integrating them. Okay, but then when we got this data, our collaborator told us that they didn't find any cell type specific disease signals, which is not surprising because your data is single nuclei versus single cell RNA, different protocols. They're quite different. I mean, and they're looking for disease specific signals, which is weak. Okay, so our goal was to improve upon these methods. Okay, so in order to improve on these methods, let me just give you a quick overview about how these methods actually work. Okay. So let's look at two of the most popular methods like Harmony and Seurat. Okay, so what Harmony does is, these data live in very high dimensions, right? And there's many samples as shown by the color in this plot. The color represents the sample. So in single cell data, each sample is a batch of itself. Okay, and then they're sort of the symbol of the markers is the cell type. Okay, so our goal is to sort of merge cells from the same cell type, so merge the colors together within each symbol type. So what this method does is it first soft assigns cells to clusters. It applies k-means clustering, but it adds a term in the penalty that favors mixing of data sets within each cluster. So it sort of has this entropy within cluster entropy term in its objective function. And then after it got these clusters, it gets the centroids of each data set. And then it pushes the centroid for each data set into the cluster center. And that's supposed to get rid of the batch effect. Okay, and then it moves the cells within each data set based on the soft cluster membership. So it's sort of like a massaging sort of algorithm. It's sort of, it has this penalty for how separate the batches are within each cluster, and then it sort of clusters to minimize that penalty. And then it sort of does this sort of prod the cells to be in the same cluster, prod the cells from different batches in the same cluster. So that's what Harmony does. Okay. Another method, Seurat, okay, which is very popular. is the one that has the most citations, like 12,000 citations. Okay, so this is a completely different method. What they do is they first apply canonical correlation analysis. So you have the same genes across different data sets. You sort of, canonical correlations, finding the best projection of cells to merge the genes together. And that puts them on like the same embedding. And then on this embedding, it tries to like find what they call anchors, which are cells from different samples that are mutual nearest neighbors of each other between data sets, and in this common embedding. And then it tries to like merge them together based on these anchors. So I don't have time to go through the details of these two methods. Okay, and then there's many other methods with different ideas in this field. So it's a very active field of research. But I just want to give you a flavor. I just want to say that all of the methods that we tried, they do what we call data massaging, which means they don't really have a model of how batch effects affect these samples. And they sort of just tries to merge, like use penalty functions to penalize for batch differences and try to just, put them together. They don't have any, they ignore the experimental design. Now, what do I mean by experimental design? Let's go back to the kidney example, okay? So here, they actually included 17 control samples, which are not supposed to have any disease-affected cell types, okay? And the question we asked it was, how were these controls used during the integration step? They were not, okay? So in the integration step, they were ignored, and basically all the samples, all 36 samples were just massaged together so that the cells are together in one cluster, hopefully in the same cell type. And the control versus disease status is only used after the integration as a baseline for comparison to look at differential expression. Okay, so what we thought was, maybe we can use this experimental design, not merely as a post-integration baseline for comparison, but also to learn about the unwanted variation in the data. Here's another example. Okay, so this is a longitudinal study conducted at UPenn, where our collaborators wanted to find the effect of a drug on the T cells, the CD8 T cells in the blood of these patients. So they had 10 patients that they treated with a drug, and then they assessed them longitudinally, and then took blood samples to be sequenced, and they sorted the CD8 T cells and sequenced them. And the goal is to identify longitudinal differences in the cell types, and then to compare different treatments. But then so if you were to integrate, again here, if you were to just integrate all the cells together across time, across treatments, you're not left with very many temporal or treatment differences. However, what is ignored by these integration methods is that there are the samples at time 0 where you do not expect any biological differences, and they can be used to learn about the unwanted variation. Okay, so with that setup, I just want to, sort of put this sort of mathematical. It's not a very mathematical talk, but I just want to give you a sense how this looks like as matrices. So we have different samples, and then from each sample we get a matrix, cell by gene. The cells are all scrambled; they're just sampled from the tissue. And here we have the same features, so same column labels. Okay, so some of these samples don't really differ from each other, by the standards of the study. If the study is standard, if the study is looking at disease, these are maybe the healthy people. Okay, now, so what we proposed was that let's give them a matrix decomposition, where each matrix, each cell by gene matrix can be decomposed as a cell state embedding. Okay, so a low dimensional embedding of the cells multiplied by the sum of 3 terms, a main variation, so the M that is shared across all samples, and then the batch variation, the remaining variation that differs across samples. However, the batch variation has the same latent space across all samples. Okay, and so how do we, so okay, I want to just clarify that we don't know any of these. We don't know C, we don't know M, we don't know V, we don't know W. And then these sample-specific left score matrices are also unknown. Okay, so what is the algorithm that we use to estimate these unknown quantities? So this is what we proposed, okay? So first, we will use existing integration methods to align the cells. Granted, that's very aggressive, right? It's gonna remove a lot of the biological signal, but it does give us a useful low-dimensional embedding where all the cells sort of can be comparable. We stack this low-dimensional embedding and get the SVD, and then we throw away the right singular matrix, and then this we will take for now as our cell state embedding. Then we go back to the original raw data, which has a lot of the lost biological signal, and then we regress on our cell state embedding to get right regression matrices. Now, these right regression matrices not only contain the biological signal, it also contains the batch effects that were supposed to be removed, right? So what we do is we average them first to get the main cell state variation, subtract it out, and then stack these residuals across only the control pool samples, and then perform SVD on that to get our V matrix, which is the latent batch latent space. And then we share that across all the samples in the experiment, project out that variation from these residual matrices that we got halfway. And then we add that back to the M matrix, which is the main embedding, and then multiply it by C to get our batch corrected feature matrices in the original feature space. Okay, so comparing what we have now versus what we had before, this is what we would have, the biologists would have ran off and started analyzing before, and this is what we propose that they use instead. Okay, so does this work, right? So we have to convince not only our collaborators, but also ourselves that it actually works. So the way that I feel that was one of the hardest things actually in this study is how do we know whether it works? We didn't want to rely on simulations or asymptotics or external validations, which is incredibly hard. We just want to know on this data set that our collaborator gave us that they spent a lot of money to generate, does it work? Okay, so this is what we call internal validations. So validations on the data that you actually care about. It's kind of like cross-validation or conformal prediction, that kind of concept. But this is not a supervised problem, right? So how do we do internal validations? We ask 3 questions. First of all, did we recover the biologically relevant variation, right? Did we improve the signal? Did we keep batch effects out? Because we added back variation that was supposed to have been removed in the integration. So did adding back that variation also add back batch effects, which would not be ideal? And then also, did we reduce the data distortion that was caused by integration? So I just want to show you some, the gist of what we did, okay? So in terms of whether we recover biologically relevant variation, it's really hard to see if you were to just try to visualize it on clusters, okay? So for example, here's a harmony versus CellANOVA integration. You can't tell, okay, based on these low dimensional embeddings. So what do we do? So what we do have from our collaborators are various sample labels, like is this treatment 1 versus treatment 2? Is this disease versus normal? So based on these labels, what we could do is for each cell, identify its K out of batch nearest neighbors. So the K nearest neighbors in this low dimensional embedding, that not the UMAP embedding, like the K dimensional embedding that's output by the integration, usually 50 dimensional, 100 dimensional, like that. So we in that embedding we get the K nearest neighbors that are out of batch. And then we asked whether they are enriched for cells from its own condition. So if it's a disease cell surrounded by disease cells, okay? So if we did that with the kidney atlas data, which is what they are trying to squeeze some signal out of that. So we can plot out the proportion of the disease cell neighbors that are disease cells and ask, is this more than the neighbors of the normal cells? So orange is normal and blue is disease. You see that before we applied the CellANOVA procedure, these two densities are overlapping. And then after we applied it, they separate, which is good, right? That tells us that these, okay, so each row is a different cell type. That tells us that these cell types are disease relevant. Now, well, that was not enough to convince our collaborators. She wanted us to do a replication. So we went to another consortia. There were two competing kidney consortia that also did a similar study, but they only used single-cell RNA-seq, not single nuclear RNA-seq. So that was easier because it's all the same protocol. So we applied our method also to that. And you see that the same cell types also had an increase in separation after the CellANOVA. So that sort of convinced us that we are onto something. Okay, so CellANOVA recovered disease-impacted cell types that are replicated. Okay, however, did we also add back batch effects, which would not be ideal? So what we did was we did a leave one out study where we took the K control samples, in this case, I think 17. We left out one treatment sample, and then we asked if we use the others to estimate the batch effect and try to remove it from the treatment sample, we will get it to align, right? So what is plotted on the horizontal axis is sort of a measure of mixing, and you want that to be as high as possible. You see that before you do the integration, it's very low, and after you do the integration with Harmony, it increased, which is good. And then after you did it with CellANOVA, it also stayed about the same, which is good, because that means that you recovered signal without reintroducing batch effect. Okay, data distortion is a different consideration. Let me just show you this plot first. So here is a plot of p-values before and after batch integration. What this means is that, so the p-values are differential expression analysis between cell types. Now, if we take cell types within batch, so we take one sample and compare different cell types, there's no batch effect there because they are from the same batch. So we use that p-value as a gold standard. Now we look at the same cell type after it has been corrected for batch, right? So we plugged it to the same machine and did batch correction. And then we asked after batch correction, did the p-value, did the within batch p-value change? And you see that it did change a lot. It became it exaggerated after batch correction, which was a common phenomenon that was people in the field sort of see that they can't trust the p-values after integration. But then after you do CellANOVA correction, it removed that bias. Okay, so basically, because of this, now we have a way of integrating the cell types without introducing these distortions, and also recovering a lot of cell type specific pathway, disease-relevant pathways. So these are 4 cell types that my collaborator felt were indeed, based on her intuition, was biologically relevant. And you see that with the signal recovery that we introduced, we were able to identify pathways that are also replicated in the independent study. Now, we wanted to publish this in a journal that biologists read and care about. So, we wanted to do a validation. How do you do a validation? So we appeal to the newer spatial technologies, because in single cell sequencing, the reason why there's so much batch effects is because you have to sort of pull apart the cells, dissociate them from tissue, and that process introduces a lot of stress to the cells. Okay, so in spatial technologies, all you do is you do a slice, you cut a slice. That doesn't, that's a different kind of stress, still stress, but if we could repeat this same sort of do a single cell and do a spatial experiment and find the same signal, that sort of increases our confidence in it. So we took, the top signal here, this specific pathway, which was not identified without signal recovery. And then we looked at it on spatial transcriptomic data, and we see that this pathway if you look at the injured versus normal proximal tubal ratio, which is like the disease versus normal, you see that they light up in the same regions of the tissue as that ratio. So that sort of gives us confidence that these methods do give you biologically relevant signal. Okay, so I know that is quite a sort of quick overview of this method, but sort of give I want to. My goal here is to sort of show you the two extremes of single cell integration, and I want to go now to the second problem, which is one of weak linkage. Okay, so many people thought this is a very hard problem because you're sort of basically trying to integrate, remember, two modalities that really have very little overlap. So one is single nuclei RNA-seq, one is spatial proteomics, and usually there's only a few, like 30, 40 proteins that are overlapping between these modalities. Okay, so it's difficult for a different reason. Here is a weak linkage, but you are not dealing with a lot of samples, and you're not trying to find disease-relevant signal. Okay, so I want to just show you some of the key ideas that went into this pretty complex pipeline that we developed to do this analysis. Okay, so the setup, in matrix format is that you have, let's say, just two modalities. You have RNA and protein. Okay, the rows are the cells, and the column is a feature, and they have different features. And one is very narrow. One is narrow and very narrow because you don't have that many features. And then here in blue are the shared features. So for each protein, we can identify the gene in the genome that codes for it, and then we can measure its RNA, right? So there's these linked features. The problem here is that there's weak linkage, right? So the existing methods before we developed our method basically use these linked features and applied harmony or CellANOVA. Okay, you see, it's not using the unlinked features because there's no way to really align them. They're different cells, different features. What can you do, right? So, well, the intuition we brought here is that even though these unlinked features are not shared, they're still useful in helping you denoise the linked features because the data is very noisy. Do I have that? Okay, so so the data is very noisy. So if you were to just align the linked features, you just, they're so noisy, you don't see much alignment. Okay, so for example, here is a method that we developed many years ago, where we try to do this kind of denoising, where we use all the genes in a cell to improve the expression of those, a subset of genes. And you see that before denoising, there is very low correlation between protein and RNA, okay? However, and that is improved a lot by the denoising procedure. Okay, so I don't have time to tell you about the denoising, but I can just tell you that got us to first base in this problem. Okay, so the first thing we do is we use the unlinked features to construct this graph, and we use graph smooth denoising to get a better set of linked features. And then after that, we use the linked features to sort of find anchors between the samples. And then using these anchors, we map all the samples onto the same low dimensional embedding, okay, by CCA, by canonical correlation analysis. And then on this low dimensional embedding, we again apply this denoising. So this is just a loop, okay, where we did anchor identification, co-embedding, and the denoising. So iterate that a few times, and then we're at second base. And then after that, we just, because not all cells can be matched between data sets, because sometimes, you have a cell type that is just lost during single cell dissociation. So what we do is we identify the pivots, and then we use that to propagate the alignment, which I'm not gonna go into. Okay, but I want to show you what a tremendous difference this makes. So what we did was we identified a data set where it's called Psych-seq, and what Psych-seq is, for every cell, it measures both the RNA and the protein. And for this technology, they measured 228 proteins. So that is a lot, okay? So if you were to use 228 proteins as the linked features to align the data sets between RNA. So remember, for this technology, we know the truth. We know because for each cell, we have both the RNA and the protein. So you have both. So you know how they are supposed to be matched. So we have the gold standard. If we were to sort of pretend we don't know which cell this protein goes with, so if we just apply these methods, we see that both our method and Seurat, which was the state of the art, do pretty well in the sense that they were able to align the modalities and identify the main cell type clusters. However, if instead of using the 228 proteins, we only use 30 most informative proteins, which is what these spatial technologies are using, they're only having 30 or 40 proteins, then you see that, without using the unlinked features, you completely mess up, okay? So it's not able to align the cells, identifying the main cell type clusters, whereas we still do a decent job in the sense that we still were able to identify the main cell types. Okay, so there's other validations we did in our paper, but I just want to show you this one example, which I thought really convinced us that this kind of cross-modality integration is able to give you a whole that is better than the sum of its parts. So here we're looking at a tonsil with, a lot of immune cells in the tonsil, and we're sequencing those immune cells using RNA-seq as well as spatial proteomics. Okay, so here you see the two modalities, protein and RNA, on the same embedding. They are overlapping, which is great. And we were able to find the main cell type clusters we expect to see in the tonsil. But then in the tonsil, we know that there are very subtle differences that are spatially associated. We know that these are the B-cell germinal centers in the tonsil. And we know that as you go into the germinal centers, you expect to see different gene expression signatures as if you go out of the germinal centers. So because we have the spatial locations, we can plot the expression of genes that were not measured in spatial proteomics. So these genes, you only know their spatial location because you did the integration of spatial proteomics and RNA, that we can now plot the expression of these genes to see how they vary in space. And we separated the genes based on existing biological knowledge about how they should be varying. And you see that they do vary in the way that we expect. So this is proof that by integrating two technologies, one where you have the spatial coordinates, but only a small set of proteins, and the other where you have the whole transcriptome, you are able to actually get good estimates of genes that you didn't measure in the space. You can get measurements of them that are pretty accurate. Okay, so back to this colon atlas that you saw before. Now we're able to identify, we can basically have protein, RNA, and chromatin accessibility in space at high resolution. And that allows us to be able to study the full gene regulatory hierarchy in space. Okay, so just a summary. So I gave you a tour of what single cell integration is about. You could have many different, these are two of sort of extreme integration scenarios. There are many other problems that fall in between, and they require different mathematical models. Okay, and I still think this is a very exciting developing field in need of rigorous statistical mathematical treatment. And I just want to thank my collaborators and students who did a lot of the heavyweight lifting in this work. Jane is the first author of CellANOVA, and Shu Xiao and Bo Kai are the first authors of MaxFuse. And I want to thank my collaborator, Zhong for all the intuitions he brought to this problem, as well as our collaborator, Gary Nolan, who generously shared with us the data, as well as all these other wet lab collaborators who helped us with validation experiments. So thank you.
I was thinking with the batch effects that you identified, I was just wondering how possible it would be, like you were saying, that maybe dry ice could, different levels of dry ice exposure on the cells. I was just wondering how how these processes, you've integrated so much information that are the statistical insights about batch effects then feeding back to the lab scientists to maybe come up with protocols that might somehow contain or limit or systematize the measurements in ways that would produce a batch.
That's what we're trying to do now, because before the models don't really actually estimate a batch effect. So we see them as sort of only integration methods, but not batch correction methods, because they don't estimate a batch effect. But now we do get terms in the model that correspond to how each gene affects the batch in each cell state. And from that, we were able to identify biological processes that are cell type specific, batch related processes. For example, we identified cell respiration as something that is really batch affected, and which makes sense because these cells go through like oxidative stress when they when the tissue is cut from its oxygen supply, and then so that's very common. We also see like mechanical stress and these protein, so mechanical stress related pathways, right? Which is clearly obviously, when the cells go through all the gymnastics of going through the cell dissociation and compartmentalization, that is involved. So we are now have these pathways that we know are involved. We are still in the process of figuring out which steps lead to them. But in the end, you can't really, it's impossible in these clinical settings when you're studying disease to really control it very well. So I think in the end, I don't think it's realistic to hope that we will now get rid of batch effects by a well-controlled procedure. We can try to reduce it, but there's still going to be a need for this type of integration.
Thanks for the presentation. Thank you. For the low-dimensional embeddings, we often use nonlinear to plot, like you have, but for statistical implementation, we use linear. Does it matter?
Right. So I gave this talk at an AI conference, and then because there's a lot of AI methods of the nonlinear, deep neural networks that are trying to do the same thing. So I told them that these linear methods actually beat all the deep learning methods that were state of the art, and at least the current methods. While AI methods like nonlinear embeddings, such as those, because there's a very popular method out there, SCVI, that uses variation autoencoder to identify the latent embedding. It works well, but we've tried applying such methods to estimate the batch effect. We weren't, at least our efforts, we weren't able to get very stable methods. We found the linear methods to work just as well, achieve our purposes, and they were much easier to use. This method takes 10 minutes to run on top of existing methods. So 10 minutes. So they're much more lightweight. Yeah, I think there's the work, the question is still open. How much can we beat these linear methods using nonlinear deep learning methods? And right now, I don't see it. Yeah.
That's a quick question. So great talk. The model slide for the CellANOVA, it sounds like you first, fit and then you refit the residuals to get the batch variation. But then there's one additional layer of variation, and it kind of looks like those last two stages, the coefficients might be kind of similar and maybe hard to identify. So I was wondering, if you have intuition on why you can both those stages on top of them.
Yeah,
Makes sense.
Yeah, I know what you mean. I sort of skipped over. I didn't talk about the sort of biological variation, which is I think what you mean by this third column in the matrix. So we estimate M, and then we pull the control samples to estimate a batch latent space. We remove that, and then we say whatever is left remaining, whatever is left over is what is this remaining biologically relevant variation. And so you could go ahead and continue to model that if you have like disease cohorts or temporal like time points. We haven't done that. So usually what we do is just give back this low dimension, this full feature matrix, after you add M and multiply by the C. Whether that is estimable depends on the study design, right?